Good afternoon, everyone, and welcome to this Loops webinar towards energy autonomous medical implants. My name is Martina Marisa Neviani from Velta, and I will moderate this today's session. Let's start from the very scratch. What is Loops? Loops is a webinar series that showcases EU-funded research innovation and actually creates spaces for dialogue between projects, policymakers, stakeholders around specific thematic. Today, we focus on a sort of key question, I would say. So how can we power implantable medical devices directly from the human body in a safe and reliable way over long term, of course? That's the main question. And we will approach this question from two, I would say, complementary approaches. So from one side, from blood to power project, which actually uses triboelectric nanogenerators to harvest mechanical energy from blood flow and power a battery-free intelligent from the project. And from the other side, we have integrate project, which actually develops bio-integrated soft actuators and bio-inspired patient's own metabolic energy. So I'm pleased to give the floor to Ines and Andrea for the presentation of Blood to Power project. After their talk, we will move to integrate project with Alessandro and Michael. And then we will follow with a joint discussion, a Q&A session. So I really invite the audience to write the question, and then we will read them at the end, and we will give some answer. Thank you all for joining, and now it's my pleasure to give the floor to Ines and Andrea for Blood to Power presentation.
Hello, so I'm Inês Gonçalves, and this is Andrea Pereira, and we're happy to join you, showing you a little bit of our Blood to Power project. So in this project, we are actually using blood as energy source to power smart cardiac devices. So this is a project, a European project led by I3S. We are located in Porto, in Portugal. If you ever feel like tasting port wine in the real place where it's made, feel free to contact us and come for a visit to I3S. So we are at I3S, we are from the Advanced Graphene Biomaterials Group, where we design, produce, and characterize new advanced biomaterials and medical devices. And then we evaluate their interaction with the host, namely with mammalian cells, bacteria, and blood components, both in vitro and in vivo. And so in this project that we are leading, we are using blood as energy source to power smart cardiac devices. And as you will see, we are using it to create a new implantable device. So we are part of, this is an EIC Pathfinder project, and we are part of a challenge, which is towards the healthcare continuum technologies to support radical shift from episodic to continuous healthcare. And I think on this, all of these devices that are powered by alternative energies will have a very important role. So our consortium is composed by ourselves at I3S, by here in Porto, by our partners that are located also in Porto in the Faculty of Engineering, which have expertise on energy harvesting, with our partners from San Sebastián in TECNOM, the Technical University of Navarra, that are specialized in electronics, and our partners at the Medical University of Vienna with a specialty in medicine. So together we make this Blood to Power consortium. So probably you know that cardiovascular disease are the deadliest problem on Earth, killing one person every 2 seconds. So this actually occurs when we have a blockage in our arteries, and this can occur mainly either in the heart or in the legs, in the limbs. And when it happens, we need to perform a surgery called the bypass surgery, where we use a vessel or a graft to go from one part of the heart where it's the flow is still occurring to another one below to reestablish the blood flow to all that area. So the gold standard is to use autologous grafts, which are grafts harvested by the person and use them in these arteries that are blocked. But they are not always available. They need to be harvested from somewhere else, which is obviously not good. And they have associated comorbidities, such as infection. So due to this high failure rate leading to death or amputation, it shows that we have a really huge need to real-time monitoring of these vascular grafts performance. So they end up saving people, but at the same time, we know they will fail in a matter of time. So if we think about the medical devices of the future, including the vascular grafts, of course, they should actually be able to sense and monitor its performance and to telemetrically emit data or alerts to the medical doctors or anyone in a healthcare system so that medical actions can occur to avoid the failure of these grafts. So the big problem in the implementation of this Internet of Medical Things is that we still have a big problem, especially in implantable devices, which are the limited use of batteries to power them. Because the batteries that nowadays are used, they're usually very large or bulky, they have very short lifespans, and they may release toxic components. So this is really not possible to use in many devices, such as, for example, the vascular grafts.
And here emerge where we can harvest this energy. So in our body, we have different sources, namely, for example, the thermal part, where we can build an energy harvesting system that use the thermal gradients within our body to produce electrical power. But if you think about the thermal, we have some disadvantage because our body temperature is really high regulated, and it's not so possible to find these gradients of temperature inside. Then we have the chemical part because our body has the metabolites, and because of this we can also harvesting the energy, but you will learn more about this with our next speaker. And our project is focused in the part of the kinetics, and here we have three different types of energy harvesting, namely by the piezoelectric, where we have differences in the pressure in the materials and we can harvest energy of this, electromagnetic, and also the part of triboelectric that is the main focus of our project. And in this case, we use the motion in the body to harvest energy. And of course, that the triboelectrics that are being generated also having some problems, namely regarding the mechanical wear, because if you are thinking in something that is always in motion, we will need this part, and also the infiltration of body fluids, and that's why our project has novelty in this part. So looking for the solid triboelectrics, we have different ways of doing this. We can actually have two materials that when they are in contact together, they have different charge because they have affinity to lose electrons or to gain electrons. And because of this, when they contact with each other, they charge in a different way, one in a negative way and the other in a positive way. If you think you can put here an electrode that is only a conductive material, we can harvest electrons that are being formatted at the surface of the materials and then create a potential difference having the energy. This happens in contact separation. We can also have the sliding. So instead of materials being contact and separate, can only be friction. And in this case, we have the famous play when we were a kid, that we just rubbish the balloon in our hair and then we just pull it apart and they start to be attractive, because it's exactly this that happens. The hair gains electrons and balloon lose electrons, and then they have different charge and they are being attractive, and we can have this phenomenon. So besides this, there are different architectures of these triboelectric nanogenerators, namely using only one triboelectric material and a single electrode, and we can think about different architectures. And this led us to our eureka moment, where we thought, okay, we have the blood flow, and the blood is always flowing through the arteries, so we are having this friction in the blood and the arteries. And here was what we thought. When the blood was going through our arteries or vascular grafts, it can actually generate charge on the surface of the blood vessels. And similarly to the triboelectrics that are being used so far, we only need to put a conductive material around and then collect these electrons and generate the potential difference. And here emerged the concept of the fluid-solid nanogenerators that we will call from here as fluid tank, as a way to simplify the word. So this technology is already patented, and then we follow with the project. And not only in the blood vessels, we can think to apply this to every medical devices that have a fluid that goes through, namely, for example, catheters, stents, and vascular grafts that are widely used in our body for different applications, and especially in the ones that blood to power focus on, in peripheral artery bypass and also in coronary artery bypass. So this emerged the idea of our project as well. We have our vessels and we have our vascular grafts, and the conventional vascular grafts are here, and as Ines explained, they can occlude. They really have a high failure rate between 20 to 50%, which narrowing the vessel and leads to an occlusion that can lead to the patient's death. But with our project and with iGraft, we need to give features to these conventional vascular grafts. And instead of being only a vascular prosthesis, we will adapt to be self-powered by by harvesting the energy of the blood flowing through the vascular grafts, and also try to correlate this output to have a narrowing sensor and have energy to send the data telemetrically to outside of our body to tell the status of the vascular grafts. And with this, if we detect a narrowing in our vessel, instead of culminating with the patient death, we can in fact send an alert for the medical or the provider or the hospital, and with this prevent a second event and not being safe here for the patient. So the objectives of the project are in fact to validate this technology, the fluid tank technology with the blood flow, and also explore the usage in intelligent vascular grafts as narrowing sensor and send this telemetrically for outside of the body. So our iGraft, we intend to have the first battery-free intelligent vascular graft that is not only a vascular prosthesis, but is a energy harvesting system and a narrowing sensor that can monitoring the graft performance 24/7, and also that can emit an alert wirelessly to the medical care. So I will show you the results that we have here. We start to do this in vitro with mimic the blood flow, physiological blood flow. And in fact, we were able to harvest energy in this way. And we have also a correlation between the flow rate and also the output that we generate. If we have a higher flow rate, we can generate more energy, and with this having more power in the end. We also use this in synthetic vascular grafts and also human vessels, because some of the vascular grafts are also autologous, so we were interested in this part. And we can see that we can also generate energy not only with synthetic materials, but also with human vessels, and can reach outputs up to 7 volts and 10 volts for the synthetic grafts. We already start our setup in vivo, validating the results in vivo if we can generate energy from the blood flow. And for this, we use the porcine model, and we did it in live native vessels of the animal. And we can see that we have peaks here, and we can generate energy from that. And also using synthetic vessels, we also validate this in vivo. So what can we do with this energy is also what we were curious about. So we can already storage it, so we can use it anyway afterwards. We can storage in a capacitor. And we can also use it as a flow sensor. And this part is important to develop the eye graft, because we have a correlation with the blood flow and the electrical signal. So like this, we have a self-power sensor. And also, as proof of concept, we can light up some LEDs and use it and visually see the energy that we can produce. So, of course, as Inês mentioned, this is a consortium. We have people from I3S, University of Porto, Tecnum, and Medical University. We have also clinical advisors with us that help us to tune the eye graft and to turn it into a clinical solution in the future. So, thank you for your attention.
Thank you very much, Ines and Andrea, for, I would say, this very clear and inspiring overview of your project and also of the eye graft concept. Actually, it's very, very interesting for us. I would say that I would like to avoid speaking right now, because it might be very interesting understanding how your thematic with the one of integrate can be, let's say, linked. So I would like to immediately leave the floor to Alessandro and Michael for their presentation, so that we can have a final talk and question at the end.
Okay. Hi, everyone. Can you hear me?
Yes.
Okay. So I will share my screen. So hi, everyone. I'm Alessandro Ianiro, and I'm the scientific coordinator of the Integrate project, which is also an EIC Pathfinder and is co-financed by the European Union and by Switzerland. So we, let's say, we defined this project, we conceived the project with a similar idea to Blood to Power, but we had in mind a more power-hungry application, which is robotic prosthetics, for which typically you need, let's say, larger amount of energy, and therefore we, as you will see later, we try to target a different type of energy harvesting. So the idea is that, there are about 30 million people in the need of prosthetics in the world, and practically, let me hide this. Practically, the most advanced prosthetic devices, which are robotic devices, they are typically very expensive. They are hard, so there is a big mismatch between their mechanical properties and the mechanical properties of human tissues. But the bigger problem is that their use is always bound to the availability of electric power, meaning that you have to recharge them continuously, and also you need to have electric power available. So this might also have, let's say, an effect on the geographical distribution of this type of devices. So the idea behind the Integrate was to practically work on two technologies in parallel. One was to define a new way to make soft prosthetics, developing a way to developing new artificial muscles that can either integrate it within the body or integrate it into soft robotic prosthetics. And I will not speak about this part because here we focus more on the energy harvesting. But the idea is that with the technology we have in mind for the soft artificial muscles, this muscle should be really power efficient, and therefore we think that we can, in principle, power them using another source of energy which is inside the body, which is not really a chemical energy, but is a form of energy that the body uses to, which is called ion concentration gradient. So in our cells we have pumps that pump practically sodium and potassium in different directions and create a concentration gradient. And this gradient is a source of energy if you can kind of harvest it. And similar things happen in other parts of the body, like for instance in the stomach. So our idea was, why don't we create an artificial electric organ that can harvest this energy and use it to power artificial muscles. So before I give you a bit of details, so the project structure, the consortium involves 5 partners. The Adolf Merkel Institute in Switzerland, where the bionic muscles and the electric organ are mostly developed. And then there are CNRS and Eindhoven University that support the experimental activities with theory. And we have also a university from Roma, Tor Vergata, which is actually helping us with more engineering tasks, for instance, 3D printing the artificial muscles, and also designing, a bit the device for the electric organ. And then Velta, which is practically the official coordinator and manage the relationship with the EU, but also the communication with the public. And our activities are overseen by a board that includes Merck and Medtronic, which are 2 big companies. Merck is more interested in the artificial muscle part, and Medtronic more for the electric organ. And then we have 1 doctor and 1 surgeon that help us also with the medical aspects of the project. And I also added KU11 because I recently moved here, and now part of my group is also contributing to this research. So how do we want to harvest energy from the body? So as I mentioned, we want to use ion concentration gradients. And how do you harvest energy from ion concentration gradients? Typically, you need to use a membrane that is selective for some of the species that are there, and practically that they can channel specific ions, in this case, in specific directions, creating electrical currents. And this is very well known. And it's used in different types of contexts, really from sensing to energy recovery from water streams. But what we want to do is do it inside the body. And we are following 2 main strategies, one involving commercial membranes, because this is, of course, more a faster track to commercialization. And then we are also developing in parallel a different strategy that really mimics the membranes of cells, so really using a bio-inspired approach. And in a few minutes, Michael Meyer will give you a better overview of the strategies, especially on the bio-inspired ones. But let me just say that so far the strategy that we found more promising is to let's say target the pH gradient that is in the stomach. You know that our stomach produces gastric juice, and the gastric juice are very acidic, means that there is a high concentration of protons in there. And of course, for our stomach to avoid self-digestion, is creating a kind of a protective layer. And this protective layer is pouring protons, so there is a proton gradient between the inner part of the stomach and the wall, which is quite steep. It's 6 orders of magnitude, more or less. And we are trying to really harvest this gradient. And so far, the first implementations were not extremely successful, so we could get to microwatts. But now, the latest prototype, which we are still validating and developing, we expect this to get in the ballpark of 10 milliwatts. So this is already a significant power output, which might be enough to do, for instance, to power a pacemaker or other type of small implantable devices. Of course, there are many open questions, and we have to slowly deal with them one by one as we get closer and closer to application. For instance, fouling of these membranes, the type of electrodes that we choose, the surgical approaches to really implant this device later on. And these, of course, are just things that we can discuss in a few minutes. But before I move on, I just want to thank all the team. So you can see here in the picture, both Michael Meyer and I are from the Adolf Merkel Institute team, but here you see also the main partners. And with this, I would like to hand the floor to Michael for the second part of the presentation.
Excellent. Thank you very much, Alessandro. Yes, my name is Michael Meyer. I'm talking to you from Switzerland here in Fribourg. And let's see, let's see, how do I, here, sorry, yes. Okay, yeah, you should hopefully see this now in full screen mode. I hope that's the case. Alessandro, can you tell me real quick? Do you see the full screen? Yes. Okay, excellent, thanks. All right, so I will talk to you a little bit in more detail about these bio-inspired artificial electric power sources. Maybe the most interesting part of this presentation might be the actual natural inspiration for this work. And you see here two animals you might have heard about. On the left side, the electric eel, who's actually not an eel, it's in really life, it's a knife fish. But you can see here on the bottom, these animals are able to create stunning electrical outputs of very high voltage and considerable power at peak. And then we have one that's even better at generating power. That's the electric ray, torpedo ray. And these are saltwater creatures that can produce up to 1 kilowatt at peak output. So very significant electrical output. And the cool thing about this is that all of this power is, of course, generated within the constraints of biology. This is all just from salt gradients, and there's no metal electrodes involved. So this can be done. I think that's the main important message here. There are biological systems that are able to generate very significant powers, at least for short periods of time, in a pulsed way. Okay, so our goal in the group for the next 5 to 10 years is to develop an implantable artificial electric organ that recharges using metabolic energy. It's, of course, one issue is to keep these devices going as these animals are able to do, and that's a big challenge that we're working on. But I would like to give you a little bit of an insight of how this power is generated in, for example, the electric eel here. And for that, we can zoom into the so-called electric organ of this animal. Maybe one thing that's interesting is that 80% of the length of this animal are composed of the electric organ. So this is essentially mostly an electric organ with a little bit of fish around it, if you want. And you can see here in detail the arrangement of electrically active cells, so-called electrocyte cells, that are arranged in series. That's important, so that the small voltages are additive. Arranged in series across the length, so up to 6,000 of these cells are in such an electric organ behind each other, and then in stacks with insulating tissue in between, here shown in purple. And if you zoom in and look at one such electrocyte cell, you can see there's an extracellular space that has a high sodium concentration and an intracellular space inside of the electrocyte that is high in potassium concentration. And then there's membranes, like Alessandro mentioned, that are selective or can selectively be opened to let sodium ions diffuse into the cell and potassium ions to diffuse out. And when that is triggered by a nerve signal, then this movement of ions generates a so-called transcellular voltage of 150 millivolts. So there's a voltage across one such cell from the left red compartment to the right red compartment. This repeats at the same time in a synchronized way over the entire electric organ by a nerve signal. Okay, and so then how could one possibly mimic such an electric organ? And we chose to explore this with hydrogels. And what you see here is an array of hydrogel lenses. These are so-called precursor solutions that you can then cure to form so-called hydrogel lenses. And if you make two such arrays that are complementary, and in red you have a high salt concentration, in blue a low salt concentration, in green a cation selective hydrogel, and in yellow an anion selective hydrogel. And if you bring those together in this motion here, and that essentially mimics the nerve signal, it brings everything together in one swift motion, and then you can set up these electrical potential differences inspired by the fish that then across every 4 of these hydrogel lenses generate about a voltage of 180 millivolts, and that is then additive through the sequence of this device. And you could see here we generate 110 volts using 2,500 cells in this way. So that showed that you can actually generate significant voltages, but one problem was that this long arrangement of hydrogel lenses led to a very high internal resistance. And that's one thing that the electric ray, the torpedo ray, solves much better by making flat cells with higher surface area. That's shown here. And we then went on to mimic that arrangement by using a paper that was soaked in gel. So we obtained a material that was easy to handle and very thin with high surface area. And by stacking these, you can now see we can power a small microprocessor, here shown on the bottom, that black thing, that then triggers these different LEDs to be illuminated. And all the power for this comes from such a gradient battery, which you can see here on the left, also in comparison size-wise to a human thumb. You get the idea. It's about the size of a AA battery. And that can produce enough power to trigger this circuit. Problem is, of course, this is limited in time. Over time, this will discharge. And so the challenge is how to do this permanently. And that's also where Alessandro comes into play. He already showed some of this work, or at least some of the ideas. So what we did here is we explored the use of human breath. So essentially the CO2 in human breath, by blowing it into a device with the right, let's say, carbon capture solutions, you can then show that with every increasing number of breath that you blow into the device, you can set up an increasing voltage of one such cell, and then you again put those in series, as it is shown here, to again produce some electrical power that can do a little bit of work here, like illuminating an LED. So this was our first step towards using a metabolic waste product of human metabolism to power something. Problem is here, eventually you will deplete this device as well, because the carbon capture agent is not renewed. That requires high temperature to do that, and that's there's no solution to that right now. So then this goes towards what Alessandro showed towards the end, these bio-inspired membranes. This is something that Alessandro spearheaded. And you see here just a molecular dynamics simulation of such membranes. They're done in a way that they are very stable and allow us for the first time to generate very large areas of such membranes with a robustness that allows us to think about actual devices. Previously, such self-assembled membranes, lipid membranes, for instance, are very fragile membranes. They are very, very thin, 5 nanometers, and they break very easily from mechanical vibration, but also chemical. These membranes are much, much more stable because we take advantage of the interface between aqueous two-phase system, here shown on the very left. These are 2 aqueous phases that have a thermodynamically stable interface that does not mix. And we form the membrane right at this interface, and you can see all the students who help with this work. And this just shows how we make the membrane step by step. And here's then a picture. You see here for the first time macroscopically such a self-assembled membrane over an area of about 10 square centimeters, possible only because of the stabilization of the interface. And so this now makes it for the first time possible to think about using such self-assembled membranes for technical applications. These membranes we showed are self-healing. You can stitch an electrode right through and pull it back out, and every time you pull the electrode back out, the membrane self-heals and seals, and you go back to zero current once the membrane closes up again. And we can do this repeated times. This is another thing that Alessandro briefly showed. This is a device that takes advantage of these membranes to then set up a prototype of such an artificial electric organ. And this is probably a little bit complicated to see on this cartoon, but the essence is that what we illustrate here in a gray membrane, it's a potassium selective membrane. There's a blown up area on the left. And we take advantage of a carrier molecule that carries specifically potassium ions from one side to the other down a concentration gradient, so somewhat inspired by the natural system. So the red solutions are high in potassium. The carrier brings it over to the other side, and with that, a positive charge, and that sets up a voltage difference. And then we arrange this whole thing in a way that the potential differences are additive. As you can see here, every additional potassium selective membrane increases the potential difference in the expected integer way, so that you could imagine coming up with a potential difference that is high enough to do some work. Just wanted to thank one more time the funding agencies that have helped over time with some of this work and are now helping, in particular, of course, the EIC Pathfinder grant, the Integrate grant that was mentioned. And then also, of course, the students. There's Carolina Perucci here, who's highlighted in orange. And of course, Alessandro, you've already heard him speak. So that was my part. Thank you very much. I'm looking forward to some questions if you might have any. Thanks.
Thank you so much, Michael and Alessandro, for your presentation. Let me say that in projects like Blood to Power, Integrate, ideas from fundamental research, advanced engineering, clinical practice truly come together. And I would say that this mix, it's not just nice to have, but let me say that it's something that really allows us to move from something that can be abstract towards solution, let me say, that can be really, that can make the difference for some patients. I would say maybe in long term, but for sure it can be very, very interesting to have this option for some patients. I don't know if from the public we'll have some question, but maybe let me start by asking something for, let's say, breaking the ice for this Q&A session. And maybe let's start with Blood to Power, and then we will pass to Integrate. I was wondering, because some of you mentioned the fact that, of course, there are some open questions because you have made some research, you are trying to push your research on, and this is, of course, the main starting point. But right now, considering the status of your project, if you could remove one barrier today, I don't know, technical barrier, regulatory barrier, cultural barrier, I don't know, whatever kind of barrier that you are seeing right now, which one would make the biggest difference for your specific project?
That's a hard question. There are so many barriers that, but I don't know, maybe being a medical device, I guess, and being like an electronic implantable medical device, I think that a big barrier will be the regulatory part, I guess. So I think that is, but we are actually trying to work around it. So not towards it, because we are in contact with people from already some regulatory offices that are specialized in this area, and trying to already from the design of the prototype point of view, already trying to address all these parts, because we have issues in the materials part, in the electronics part, so it's everywhere. So this integration design into the final form in a way that is acceptable in terms of performance and also regulatory approval, it's hard. But yeah, I think that's mainly it.
Thank you. I think that from Integrate point of view, they will be on more or less on the same page, but Alessandro or Michael, if you want to reply.
Well, if I could remove something, I would remove the second law of thermodynamics, but that would to get much more energy. But yeah, no, besides jokes, because we are a bit more in a fundamental step still, but it is true, eventually, if this technology proves to be viable, we will end up with the same roadblocks, I guess.
Yeah, I mean, maybe just on a hopeful note, it's just very, I guess, nice to know that these animals can actually do it. It can be done. And the question is, of course, if we can do it, and there's a big question and there's a lot of challenges, but the cool thing is it can be done. You can generate significant power from gradients of salt using all within the constraints of biology. That's cool, and I think that should give us all some hope that at some point some of these things will be achieved.
So how do you see your technology maybe in 10, 15 years, or at least what do you hope for your technologies? Michael, maybe let's start with the Integrate.
Yeah, I mean, I would love personally to be able to come up with a prototype device that can really convert chemical energy into an electrical output continuously. So as long as you provide food, for example, in the form of lactate or other molecules that might be available somewhere in the body, you can continue, and it doesn't have to be continuously delivering power, but at least charge some capacitor or something and then take power on demand, like blood to power was also proposing. That would be wonderful. I don't have 10 more years. I will retire before that, so hopefully it can be done before that.
Thank you, Michael.
We will still be around. Andrea, do you want to?
Yeah. In case of blood to power, I think that what Ines said about the barrier of the regulatory, it's really a barrier to put the device in the market. But the technology, I think that is quite simple in a way that we have the blood there, we already have the vessels, the vascular grafts are also commercially available and already approved from the FDA. The only thing that we need to do is to add on a device that is coupled in the outer part of the vascular graft, so it will be not in contact with the blood. So I believe, and maybe it's a dream, but that in 10, 50 years we can see the eye graft in the market, and also different devices that are smart, because we are going towards this within our human body, because we already have smart house, smart cities, and smart health is just coming, and I think
the
that's
natural
now,
step.
yeah, it's the era of these devices. And the energy harvesting is one of the big issues, and we are addressing from this part, but also all the other types of energy harvesting, if it starts to be efficient, it will boost this kind of devices go towards something that is intelligent and that can change the paradigm in the medical devices.
Thank you so much, Andrea. I saw from Alessandro and Michael that there is a sort of agreement there. So
yeah.
Thank you so much. I cannot see a question from the public, but maybe let's somehow close with something that is much more near to the patient point of view. Do you think that from their perspective, they might need some sort of specific kind of communication to better understand what is the impact of those kind of researches, or do you think that it will be easier from their perspective? So much more difficult from the medical point of view, from the energy, construction point of view, but it will be easier at the end, so with the patient, or do you think that we will have some barriers also there?
Yeah, that's actually a very good question, and we all wondered about that. And we actually have a specific work package in our project dedicated to that, because we had like a survey and interaction with people around to understand what is their actual knowledge about this Internet of Medical Things, and trying to understand if they have a medical device that can send alerts. Who should those alerts be sent to, if they want to know it, if only the medical doctors should know it. So, and actually most people don't really know what these kind of devices are, and they are still a little bit afraid of, we ask them, so what would be the main concerns, for example, and so they still have some concerns regarding the data protection, and I don't know, Andrea, do you remember? So there are, sorry, it's a huge amount of data that we are currently treating. So, but I think that's a very good point, and I think that our population is not ready at all to start using it. But now more and more we see the smartwatches, and then the whoops, and so I think that more and more devices are coming, and of course people use them. Most of them are still wearable, so not implantable devices. But I think that people will start getting used to them, and then when the time comes to use the implantable ones, probably it will be a little easier. But I think that it's also our job to highlight these points and also educate the population on these topics.
Yeah, maybe I can just add that, in fact, people, when knows what Internet of Medical Things are and smart implants, they said that they want to have it. So we are in a good track, and people, after understanding what it is, they want to have this on their devices. So
yeah, I guess at the end it will depend also on how invasive is the procedure compared to what is the alternative. For instance, for pacemakers, replacing a battery is invasive, yes, but it's every 8 years, 10 years.
Yeah.
So if a person that is 60, 70 have the choice of replacing a battery once in a lifetime or some other more invasive procedure, they might not agree. But if it's less invasive, then for sure, so this.
But it may be equally invasive, and they have the chance to know if it's failing or not, or if it needs to be charged at some point. Exactly. I think the idea here is to make it more so the people outside know exactly what is happening in the device.
Indeed.
And